칼날이 위쪽, 칼을 거꾸로 잡았다?
From the second a crime is committed, traces of the crime are left in the environment. The location where a crime took place can be full of physical evidence, which can link the crime to the criminal and ultimately assist the police in their investigation. If you ever wonder how the forensic unit uses these pieces of evidence to trace the criminal, be sure to keep an eye on this video till the very end. What is DNA? DNA stands for deoxyribonucleic acid, made of chemical building blocks consisting of 3 parts, a phosphate group, a sugar group, and 4 types of nitrogenous bases called nucleotide. The nitrogenous bases are of 4 different types: adenine, guanine, cytosine, and thymine. Adenine will always bind with thymine and cytosine with guanine. This type of bonding is hydrogen bonding. Phosphate and sugar are linked together to make the backbone, and complementary bases are attached to it. Due to the bond angle of sugar-phosphate molecule, the linkages will eventually form a double helix structure. So, the question is, how DNA helps to identify the criminal? By the materials, fingerprint, and blood at the crime scene. Let's look at them one by one. The example of materials are shirt, sharp weapons, hats, or anything at the crime scene. Other than that, human leaves fingerprints on things they touch. Research shows that DNA can be obtained from a single fingerprint. A forensic scientist can also use blood evidence to contradict a witness statement or any statement that the suspect may make. Hence, it can help determine if a crime is committed. Wait a minute, let me introduce you to Jamie, a forensic specialist. Jamie!
Hey there, I'm Jamie. I'm here to clear a misunderstanding and talk about what forensic can do, which turns out to be pretty interesting all by itself. And to do that, we are going to solve a hypothetical crime. So, here is our case. Someone broke into Jenny's house last week. The police can identify the suspect from blood found at the scene. How can they do that? To understand this, we need to state what is meant by DNA fingerprinting and describe some uses of DNA fingerprinting. The burglar smashed a window, leaving blood on the broken glass. Samples of blood, hair, or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzyme to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electron current. Different size fragments travel different distances in the gel. The smallest fragments travel the farthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzyme cut different people's DNA in other places, creating a different set of fragments. The different stages of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA, which will produce a unique pattern that can be used to identify them. If 2 patterns match, the samples are from the same person. So, the police can identify the man who robbed Jenny's house using his unique DNA fingerprint. They make a DNA fingerprint by taking a sample of cells from someone and separating DNA fragments to create a pattern. If the pattern matches a sample from a crime scene, that person was there. By now, I hope you can understand how DNA analysis fights crime scene. That's all for me. Bye!
Today, you learn about DNA and how it can help to identify the criminal. You also investigated one robbery case with Jamie to deepen your understanding of DNA fingerprinting. I hope by now all of you understand how DNA profiles are made and have seen some example of where they can be used to detect the fingerprint and match up with the criminal. Hence, it might be easy for police and authorities to find the criminal. Thank you for watching.